When I was a kid, I played with Legos all the time. Every time it was my birthday, or Christmas, or any holiday, I would get a Lego set. And if you've ever played with Legos before, you know that each set provides a long and detailed instruction set. But being the absolute madman that I was, I almost never followed them and just winged it most of the time, resulting in pretty abysmal creations. I just really didn't care for the instructions. But you know where instructions do matter? That's right, biology. Nucleic acids, the final of our four macromolecules, program all the activities in a cell and dictate their development. Today, we'll explore the structure of nucleic acids, how they build DNA and RNA, and base pairing rules. Just like carbohydrates and proteins, nucleic acids are polymers that consist of monomers called nucleotides. Nucleotides are constructed of three parts: a 5-carbon sugar called a pentose, a phosphate group, and a nitrogenous base. Our pentose comes in two different variants: deoxyribose and ribose. Spot the difference. That's right. Right here, an oxygen is missing. The absence of an oxygen here is why we call this pentose a deoxyribose. If you want to sound really pretentious, you can call it 2'-deoxyribose because the oxygen is missing on carbon number 2. Attached to carbon number 5 on the pentose is a phosphate group. You'll notice that this phosphate group is charged, which is what gives nucleic acids a negative charge. Finally, attached to carbon number 1 is a nitrogenous base. These can come in 5 different flavors: adenine, thymine, cytosine, guanine, and uracil, or A, T, C, G, and U for short. These nitrogenous bases can be classified as pyrimidines or as purines. Pyrimidines, which are thymine, cytosine, and uracil, are nitrogenous bases that have one ring, while purines, which are adenine and guanine, are bases that have 2 rings. Nitrogenous bases are what differentiate the different nucleotides. The actual information that nucleic acids hold are encoded in the sequence of these bases. To build a polynucleotide, adjacent nucleotides are linked together by phosphodiester linkage, which is when a phosphate group on one nucleotide links to the 3' carbon on another nucleotide. In a polynucleotide, the two ends are distinct. One is an exposed phosphate group, and the other end is the 3' carbon of a pentose. We call these the 5' and 3' ends of a polynucleotide, respectively. In writing the sequence of a polynucleotide, we always indicate which end is which at the beginning and at the end of the sequence. Now, how do we build RNA and DNA from these polynucleotides? RNA molecules are simple. They usually exist as single polynucleotide chains that consist of the bases adenine, uracil, cytosine, and guanine, or AUCG. On the other hand, DNA molecules are double-stranded. 2 polynucleotides spiral around an imaginary axis, forming a double helix. These 2 strands run in opposite directions. One runs in the 5' to 3' direction, while the other runs in the 3' to 5' direction. Think of it like a divided highway. The 2 lanes are parallel but run in opposite directions. Often, this is referred to as anti-parallel. DNA contains the bases adenine, thymine, cytosine, and guanine, or ATCG. In DNA, each nucleotide on one strand is complementary to another nucleotide on the other strand. However, only certain nitrogenous bases can pair with each other. Adenine nucleotides can only pair with thymine nucleotides, and nucleotides with cytosine can only pair with guanine nucleotides. These are called base pairing rules. Here's a fun fact for you. In RNA, base pairing can actually happen within the same polynucleotide, causing it to bend into a 3-dimensional shape. Please do note that in RNA, there is no thymine, so adenine pairs with uracil. Today, we barely scratched the surface of DNA and RNA. Keep in mind, there is so much more for me to cover in this particular subject, and that you will certainly see more videos about this topic in the future. Thank you for watching, and I'll see you next time.